Good morning, everyone. Good to see a full house today for this great talk. Let me begin by acknowledging the traditional custodians of the land on which we're gathered today, the Gadigal people of the Eora Nation, and pay my respects to their elders past, present, and emerging. Look, today's talk is kind of two stories intertwined. If you think your way back to 2020, you may remember that we had a little pandemic on our hands. As chief scientist, we were asked our opinions on various things and so on. And I said, we really need to be in the position of having some sovereign capability in these areas. And at the same time, coming into the frame, Daniel Getz came to Sydney and we met and we talked to each other, and I realised he was a super smart person, an Australian, who had made his mark being an entrepreneur in the US in the RNA area. And the two stories kind of intertwined. So we got permission pretty early on in 2021 to build an RNA manufacturing facility. An RNA manufacturing facility with the difference. I was hoping to be able to show you a picture of it, but we've had a few technical pictures at the back. But a facility that basically allows anyone with any really good ideas in RNA to manufacture their materials up in this state and to attract companies that really wanted to really innovate in this space. And RNA, as I've begun to realize, the more and more I've learned about it, is a remarkable thing. It's not just about vaccines, and as you'll hear, it's also about therapies, but it goes well beyond that. You can build structures, you can build machines, you can do amazing things with these new materials. I often like to say to people, the transformation that we're seeing is biology becoming engineering. It really is a really interesting process. So we got permission to build this facility, and we started talking more seriously to Daniel. We got the formal process up and going, designs for the building, going out to tender. A number of people competed for the tender, but I was very pleased when Daniel's company won the tender to basically operate the facility. And then we also got significant funding from the government, another 119 million, in fact, basically start really putting money into research around RNA. And Parley over there from NSW has been really leading and helping us in doing that. And so the two stories got even more intertwined. Daniel here, really with the innovation component, with the ability to manufacture, with new materials, with the research that we want to undertake. We are now in a position where the walls are going up on the facility, so it will be operational probably by the end of this year, beginning of next year. And it will be a real unique facility in the sense that anyone with good ideas in RNA will be able to come along and benefit from Daniel's company's experience in manufacturing that RNA to make new products. So really is dawn of a new era. And throughout this, Daniel has been to Australia, I think, I don't know how many times you've been here. He's flown over especially to give this talk today, in fact, as well. But has been really, really valuable in talking to our researchers, in working with our universities, in really trying to get the best that we have to be corralled around this business. So it's fantastic that we've got him here today to talk about the work that he does and to tell us the sorts of things that are going to be happening in the RNA space over the next decade or so. So Daniel, as I said, from Australian. He's from the Shire. He did his undergraduate degree and PhD at the University of Sydney. And without letting out too many things, I think he decided that if you wanted to make money and you wanted to be an entrepreneur, at least in this space, you had to go to the US at this point. And so he's been super successful in starting companies and running companies in the US. He is a co-inventor on at least 100 patents in this area, though he really is a major entrepreneur in the area. And it's fantastic that he wants to give back, I think, to New South Wales and Australia in terms of starting companies like this and really helping us do these sorts of things. So I welcome Daniel today to give us a talk on RNA. Thank you very much.
It's quite a lot to live up to now. Give me a moment to get this organised. And while I'm doing that, I do want to acknowledge the fact that it takes a village or a city to do what I'm going to touch base on right now, and I'm just the lucky person that gets to talk with folks. I actually grew up in around the Sutherland Shire, a little bit in Housing Commission, And the reason why I'm often asked, why do I try and do things in Australia? And it's because Australia gave me my chance. Back when I was growing up, whether it was the cricket, rugby union, ice skating with Stephen Bradbury in the Olympic Games, it felt like if there was an Australian, up on deck to have a go, they're going to give it their best and do something pretty significant. And I think having been in the United States for quite some time now, I do feel that maybe we've lost a little bit of that, and I hope to come back and try and help maintain that motivation and, pretty much we can do anything attitude, in Australia, because it's actually true. For a country with 23 million people, we continually punch above our weight, but we can't forget that, and we can't let mediocrity be our goal. We have to continue to fight for exceptionalism. And I suppose for me, being in the United States, I've certainly embraced some of the culture. I've had an advisor for a politician of another state tell me maybe I've spent too much time in the United States. And so if I have a little bit of that culture in me today, I apologize in advance, but I think there's a lot of great things about the United States we can learn from too. So with that said, I do have some disclaimers. I do have some biases. I'm going to talk about things that I've had a pretty strong hand in developing. And so please appreciate that. This is how I pay for my 4 children to go to school and do things like that. And the excuse I'm allowed to give my wife for being here while she's managing my 8-year-old, 7-year-old, 5-year-old, and 3-year-old at home. So she deserves credit for that. And Hugh has met these folks. What's really interesting is all of these things I'm going to talk about, except for one, have emanated from what we started in 2019 at Myeloid Therapeutics. And what I think is really interesting to point out is this was all ideated within the company, right? So we didn't have any strong academic background. We had a bunch of really smart people in Boston, Cambridge that came together, and we said, look, we think we have a good idea. And we're able to convince people with deep pockets there to the tune of $125 million US to support us to do that. And what you're going to see over the next few minutes is a little of what we're doing, but also the transformational ability of RNA. And actually, my PhD supervisor from Sydney's here, Nicholas King. And what's interesting is when I was doing my PhD, I hated RNA. It was very difficult to use. It was very unstable. I could never isolate it because I was a bit of a bucket chemist. And I found a great honours student to do the work for me. So there is some irony that I'm standing here in front of you talking about RNA today. So let's move on here and just, get a bit of an introduction to what RNA is. There is quite a diverse group of people here, so I'm going to try and keep it fairly high level. And there's definitely people that are probably smarter than me in this room, so, please give me some dramatic license as I move through here to try and get everyone engaged. So RNA, it is really part of the fundamental dogma of life. Watson and Crick, who discovered the DNA helix, described this originally. And so the DNA within the nucleus is where all of our genetic code is. And RNA is really important because it's what decodes and translates that code and essentially guides all of cellular processes. RNA is translated into proteins and so on and so forth. This is very simple, right? What I've put on here. It's much, much more complicated than this, as you can probably appreciate. There's actually multiple different kinds of RNA. Today I'm going to focus mostly on mRNA and how we can target mRNA. But there's transfer RNA, there's ribosomal RNA, and so on and so forth. So it's quite complicated. What you should appreciate is RNA is pretty much involved in all cellular processes. And we're only just starting to appreciate this complexity now. As another anecdote from my time at the University of Sydney, I went to a genetics lecture in 2001. And I really enjoyed mending bar more than lectures. So if I had an excuse not to go to a lecture, I jumped on it and went and played pool and had a pint at the bar. Anyway, the very first lecture for this genetics lecture, the lecturer said 80% of the genome is junk, non-coding junk. And the lecturer actually wrote the book we were studying, so I figured if I just studied 20% of his book, by the end, I could do it. And in hindsight, that was a mistake because, like everything in biology, everything's there for a reason. And we were actually talking about this yesterday over a beer with Hugh. That non-coding region of our genome, especially within RNA, is actually really, really important. And I'll touch base a little bit on that. So what you have to appreciate is a lot of what I'm going to show you is really cutting edge. It's never been done before. Some of it is designed in America and made and applied here, and I'll touch base on that. Everything I'm going to describe today, the very first clinical application was done in Australia. I think it's really important to point out, even though, unfortunately, the ideas may have been conceived in the United States, and we can get into why that is at a time point here. So just to refocus ourselves back onto DNA and RNA to look at the very different components of it. You've all heard of DNA. It's a blueprint of life. It's stable and long-lasting. You've all watched the criminal shows on TV where they find some DNA in a glass and they're able to sequence it. RNA is not like that. RNA is very unstable, but it's flexible and it's single-stranded. It's very easy to make. So, there's definitely components of that. And RNA became famous because of COVID-19. Obviously, the ability for us to, take RNA, and what we did is we essentially just expressed a little bit of the COVID vaccine, of the COVID virus, the spike protein, and we essentially trained the immune system to recognize that. And by doing that, we made antibodies that, gave us some protection. And we can all debate about how the vaccine process went. I think we can all agree on that vaccine was essential for saving human life at a time we really didn't have a good grasp on the situation, and it certainly helped us all come back to a normal life and being where we are today. And so I think what's really amazing about the COVID vaccine, ignoring the politics and whatever your view is on vaccines, is we're able to do that in the space of about a year. I don't think in history there's ever been an example where a drug was essentially conceived and made and approved in a year. And it really goes to point out the uniquely powerful component of mRNA, the ability to work with it, the ability to rapidly design it, the ability to scale it from a manufacturing point of view. And this is really unheralded in drug development, where your average timeline is anywhere from 10 to 12 years. And obviously, there's a whole heap of reasons we got that done quickly, so I'm not going to minimize some of the regulatory and international collaboration that went into it. But RNA is actually new. It's been around for quite some time. I mean, we originally described it back in the 60s, and if you really want to go and have a look at the history of RNA, it's actually quite extensive. I think what's interesting is we've known about RNA as viruses for a long time. HIV is an RNA virus, arbovirus is West Nile, dengue, all of these other viruses are RNA as well. So we've actually had quite a bit of experience in it. And we often don't realise that Moderna actually was started in 2009. Had been working in the space for a good 11 years before they really got to where they were. And today I'm going to touch mostly on RNA in the context of therapeutics and biomedical applications. There's really no disease that you couldn't apply RNA to in the context of therapeutics. Whether it's infectious disease in vaccines, whether it's in regenerative medicine, cardiovascular disease for reducing cholesterol, metabolic disorders, for example, GLP-1 agonists. These are all areas where people are looking at. And if you look at the world for clinical trials over the last 10 years or so, you can see we've had this exponential increase in clinical trials using RNA. And so this is actually very important. And I'm actually not going to talk about vaccines at all today. I'm going to focus on everything else that RNA is used for because I think that's important. And vaccines actually don't account for much of that. But if we actually go beyond biology in the context of biopharma, the limit of what we can use RNA for is endless. And Hugh touched base on this a little bit about, structures, but in the context of climate change, people are using RNA systems to try and figure out how to engineer microbes to help with carbon and all of these things, even cosmetics. So there is so much broad application, but my focus in the next however much time I have now is to really touch base on some of the things that we're doing. So the areas where we have a strong influence is in this immune programming, where we use RNA to program the immune system to do what we want it to do, as is also in gene delivery. I did steal from a friend of mine in Cambridge some slides on using RNA to edit mRNA, and I'll touch base on that to correct missense mutations, and I'll touch base on that. And then I'll end with a little bit of what we're trying to achieve with Aurora Biosynthetics, which is the operating business of the facility over in Macquarie Park. So immune programming, vaccines are immune programming. It's just very specific to an antigen that you deliver. Now, in the context of cancer, we've really moved into a world of trying to get the immune system to recognize and kill cancer. And the prototypical example of this is chimeric antigen receptor T cells, where we take your blood cells out, you'll go sit down for an hour or so, we apheresis, we take all your cells, we find your T cells, and we actually use an engineered HIV virus, of all things. Who would have thought we would have taken something like that and made it, harnessed it for our own use? But we use HIV to encode for what we call a chimeric antigen receptor, and then that gets expressed on the T cell. We reintroduce it into the patient, into yourself, if you have a hematological malignancy, for example, and then that will hopefully kill the cancer. It's actually very good in B-cell ALL, about 100% CR rate, so it's very good in that instance. It's not very good for solid tumours, pancreatic, breast cancer, and there's a whole immunological reason for that I'll touch base on in a minute. Now, the problem is this is very complicated. The average cost to make this product is around $250,000 US. The side effects are actually pretty intense. It's about $1 million of hospital costs associated with that. And so this is really not going to be a technology, I think, that is going to be widely applicable in the future. So where does RNA come into all of this? Well, RNA comes into all of this because we can actually use RNA to deliver that receptor that I mentioned directly inside the patient. So we don't have to take the T cells out. We don't have to go and engineer them. There's no patient-specific manufacturing. And it actually costs about $200 a dose to make it. So we've gone from more than $200,000 to down to $200. Suddenly we have a very accessible, very cost-effective therapeutic. And that's what we have been focused on at Myeloid Therapeutics since 2019. So we have 2 programs in the clinic today using this approach, where we take essentially the COVID-19 components, so a nanoparticle, lipid nanoparticle, an RNA strand. On the RNA strand, we encode this chimeric antigen receptor. And we have patients that are being dosed in Australia today targeting TROP2. So TROP2 is a protein that's on breast cancer, pancreatic cancer, cervical cancer, head and neck cancer, renal cancer. There's a whole array of them. As well as we've got a program in liver cancer targeting a protein called GPC3, and I'll touch base on that as well. But what's really cool about all of this is it's based on technology that we've built at Myeloid Therapeutics. So this is everything I'm going to show you we did from de novo, essentially from a napkin sketch, and we went and do it. And I'm going to touch base on a couple of high-level things here. I could get very deep into science, and I have to avoid doing that because I am very passionate about science. But the first thing I'm going to talk about here in the product that we've made is RNA itself. So RNA, I don't know if it's going to work. This is a little pointer. Probably won't. I'll use this instead. I'm not sure if you can see this. Yeah, so RNA. We depict RNA as essentially a line. And within that line, in the middle is what we call our coding protein, what matters. And everything outside of it is non-coding, right? And what you can see here is very simple, is we essentially have 4 different RNA that express exactly the same coding protein, but we just change the non-coding regions. And what you can see here is by changing the non-coding bits, the bit I told you was junk earlier, has significant impact on expression of the protein. And this line here, this grey line, is actually BioNTech and Moderna technology. It's not very, very good, in my opinion. And the orange line is our technology that we're using. So we're very excited. And again, it points to we actually used a lot of AI to solve for this and a whole heap of different things. But it just goes to show that not only is it the protein we're delivering, but it's all of the things around it that are really important in the context of trying to get the most optimal therapeutic into the clinic. The next thing I'm going to point out, and I'm going to be actually I'm going to use this slide to describe this. The immune system has a lot of cells, right? There's monocytes, there's T cells, there's B cells. You've probably heard of all of this. Well, the most important cell, in my opinion, and a lot of people in Australia will agree on this, is what we call the myeloid cells or the antigen-presenting cells. These cells are really important. Right? These are the cells that run into the burning building. If you have an infection, you bang your knee, pretty much stereotypically, any AMI, so if you have a heart attack, the very first cell that gets called into that space is these myeloid cells, and their job is to wall off an infection or to start the process of repair. And these cells actually have a second really, really critical function, and that is to direct the rest of the immune system what the hell to do. Right? So if you want an effective T-cell response, the myeloid cells or the antigen-presenting cells are absolutely essential for that. Now, in the context of a tumour, which is in the middle here, myeloid cells have no way to actually recognise cancer cells because cancer is self. It's not a bacteria. And unfortunately, cancer is very good at co-opting these cells to help actually cancer grow. And in the context of immuno-oncology, so cancer treatments using immune-based technology, the focus has been on T cells, right? And I already talked about chimeric antigen receptor T cells. Now, obviously, this is an issue if the primary orchestrator of the immune response is myeloid cells. And so what we've done at Myeloid is focus on how do we get to solve for bridging that gap between the myeloid cell, the orchestrator of the immune response, seeing tumours. You can see here we do this using a very specific receptor. It's yellow and red here. I'm not going to go into detail on that right now, and I apologise if you're colourblind. You wouldn't see that at all. But take my word for it, that it's there. And the whole point of it is to integrate the immune response against the tumour. Right? So we're essentially bridging it here. We activate the T cells, and we do all of that. And we have a number of clinical programs. And what I want to spend a few minutes on right now, I should check time to keep on track with that, is just some of the data we're collecting. Now, I want to point out, this has never been done before, right? Usually takes 10 to 12 years to get, products into the clinic. The very first CAR T-cell product was tested in 1988, and the very first time it actually worked effectively that Carl June did in Pennsylvania was 2014, right? So we're doing something Herculean in what we're doing, and I'm going to show you some data right now that gets me super excited, that makes me feel that it's a matter of when, not if, we get these technologies to work. So I mentioned TROP2. It's a protein that's expressed actually on some healthy tissues, but overexpressed on a number of different tumour types, and you can see them here. So, we're very excited about the fact that it's broadly expressed. Now, in the context of cancer, we've come a long way, but 5-year survival rates are pretty horrible. So I'm pointing out here the most recent approved therapeutics for patients that are at the last line of treatment with triple-negative breast cancer or non-small cell lung carcinoma is only around 30%. So you have a 3 in 10 chance that you'll get a response. That response is only 90 days, right? So we have a long way to go. But this is considered a success from a commercial point of view. And so we're obviously hoping to do better than that, and we build this receptor that we deliver as a mRNA LNP, which is here. And what we do here, to be very simple, to overcome a delivery challenge, is we only deliver half of the receptor. And the other half of the receptor is only expressed in cells we care about. And the reason why this is important is one of the challenges with RNA delivery, especially intravenous LNPs, is the liver takes up a lot of them. And so what we said is, look, we don't need every cell to take up, our target cells to take this up. We'll accept that collateral damage of the liver taking it up, as long as it doesn't damage the liver. But the most important thing is that it's expressed by the target cell. And the way we achieve this is by delivering only half of the receptor. And this is actually, for this to be functional, it has to form this multi-subunit receptor on the surface. And this red component here is only expressed by our target cell. So only our target cells can express the protein. And we're in the clinic right now. We've just about 25 people across Australia. So Melbourne, Sydney, and Perth are our primary sites. We haven't had any dose-limiting toxicities. This is really exciting for us because one of the dogmas with RNAs, it's very reactogenic. If you inject it, you have this response to it, and you've probably all heard about the COVID vaccines and people having lots of different responses. And this is because people thought that RNA needed to have a very specific chemical modification, a pseudouridine modification. This is complete rubbish. It's wrong. It's been spouted by folks at Moderna because they own the patents. We have now dosed more RNA into a human than has ever been done without any reactogenicity. Our biggest problem right now is we see no safety issues. It means we have to go higher in dose to find where our dose is, which means we're taking longer to get to where we're going. Efficacy, I'm going to touch base on that in a moment right now. Actually, I'm going to skip straight to that right now. So I'm super excited about this, right? It's never been done before. Our third patient comes into the clinical trials, a 49-year-old woman, has hormone receptor positive breast cancer, lymph node and bone disease. And at her first scan at 60 days, there was a 40% reduction in the tumour mass in this patient. This patient is actually now on treatment for longer than 12 months. And on this particular scan, the way we measure responses is you have to see a tumour shrink by more than 30%. We call that a partial response. If it goes more than 80%, it would be a complete response. Now, in this particular instance, the target lesion is in a lymph node, and so obviously we can't completely remove a lymph node because it's always going to be there. So we're actually very excited by this. This is almost like a complete response in this patient. There were some other issues that they did encounter later on. But the point is that this is a patient that really was limited in their follow-on therapeutics, and now they've had this experimental medicine. This is actually a Sydney-based patient that is receiving that. And I feel very thankful that this patient put themselves into the study. So where are we now? In this particular program, we're doing a lot of work with folks in Melbourne and Garvan, actually. So at the Garvan, we're doing stuff that's never been done before again, where we take these biopsies from patients pre and post-treatment, and then we do essentially single-cell sequencing, spatial single-cell sequencing, where we can actually look at the entire tumour biopsy and look at each cell, what the cells are expressing, look where the CAR is, look where the T cells are, and Alex Sorbrek and his team at Garvan are doing that, and we're super excited. We're also using mass cytometry-based imaging done at the University of Sydney with Nicholas King, and you can kind of briefly see here the pre and post, changes in the cell composition within those tumours. So we're very excited because what we're seeing is what we want to see, which is changes in the tumour microenvironment. We're seeing recruitment of immune effector cells, so that's very exciting. We're seeing changes in the cytokine environment and also in T-cell responses. So again, this, for those of you in the audience who aren't immunologists, looks like something out of Star Wars maybe. The point here is what we're trying to do and what we're trying to show is that if we can arm this particular cell here to see the tumour, we can activate that immune response. And we're starting to collect that data. Now, we have a long way to go because this has never been done before, so we've got to figure out exactly how to dose it. We've got to figure out the duration of doses. The one thing that we've done that's also never been done is that patients had 13 doses. So every 2 weeks they come in and get an infusion. So you can safely administer RNA intravenously. Again, these patients aren't having the kinds of things that you've heard of before in the context of safety and so on and so forth. So we actually also have another program in liver cancer. I'm going to touch base very briefly on that now. And so liver cancer is actually one of the fastest growing cancers, most likely, at least in the Western population, because of our enjoyment of pints and whiskey and so on. In other regions of the world, it's the prevalence of hepatitis virus, for example. Anyway, the point is it's becoming prevalent, and we've started a clinical trial here in Australia using the same approach, but targeting a different protein. It's called GPC3, glypican-3. The very, very first patient we dosed at 50 days had a 57% reduction in their tumour mass. 57%. So you can imagine how we felt. Jackpot. Now, cancer is a formidable foe, and to really put your flag in the ground to say victory, you have to take another scan 4 weeks later to confirm it, right? Well, we definitely had victory against all the tumours that were there at the start. Unfortunately, there was one new tumour that developed that we're looking into. I don't think it expresses the target. But the point is, again, we safely administered this product. We saw an immunomodulatory effect, right? This has never been done before. It's all happening here in Australia. Right? Patients are getting access to these technologies in Australia, and we're super, super excited by that. Now, in the context, and I think this is important for people to appreciate, what does it take from a financial point of view to get to here, right? This is completely private enterprise. There is no government involvement in this context of our business. We've raised $125 million US, and we've done deals that have brought in close to another $80 million US. So $200 million to get us to where we are over 6 years. So this is a very expensive enterprise. in the context of money. So we don't take that lightly, but it's also on a scale that is moving very quickly. So, programming the immune system, vaccines, chimeric antigen receptors, oncology, fantastic. RNA, that's just one use there. I'm going to change gears to something that's completely out of my wheelhouse, but is a use for RNA that is still, very important, and that's using RNA to correct causal missense mutations. And so these are actually very common in a number of different diseases, whether it's Parkinson's disease, non-alcoholic fatty liver disease, ALS, and a number of different diseases. And what's really interesting, and we only discovered this about 6 years ago from my understanding, is within our cells, sometimes not only do you have DNA mutations, but sometimes you get mutations in the RNA code too. And our cells actually have already got a system to fix that. And it's called this adenosine deaminase acting on RNA system, where it converts amine to an inosine. Excuse me. So the whole idea of this approach of using RNA is, can you go into a cell in someone that has one of these diseases and essentially use RNA to edit the mRNA so you can take a malfunctioning protein and make it functional? And here's a very simple example, alpha-1 antitrypsin deficiency. And so this is actually a disorder. It's rare, but it results in destruction of the liver and the lungs because of a mutation in the SERPINA alpha gene. And so this particular protein, alpha antitrypsin 1, is a protein that actually helps protect the lungs from a protein from an immune cell. And when you have this point mutation, essentially you lose loss of function and you have a lot of problems within that. And so the idea is actually pretty simple, is we take an LNP, we deliver an oligonucleotide that can harness the endogenous editing machinery of the cell to correct it. And so essentially what these folks have shown is that if you do this, you can actually correct about 70% of the protein in the body, right? You still have some mutated protein, and the question is that going to be enough? And so the clinical trial is actually ongoing here. There is a competing company to this one called Wave Therapeutics who released some data recently who showed you can correct about 60% of this. So again, this is all RNA-based. Right? In this context, it's using a short RNA, oligo-RNA, to go and then harness the cell process and correct mRNA to get correct functioning. Now, pivoting back to something a bit more stable in the context of editing, obviously in that context, you're going to have to continue to dose, right? Because obviously it's a transient outcome. But what if you can use RNA to edit the genome and make stable edits that never go away? Now, this is kind of the reverse of what I told you at the beginning of what our fundamental dogma of life is, which is DNA, RNA, proteins. But there are actually many examples where RNA goes back into the nucleus and transcribes itself into DNA. So this is what HIV does. That's why it's so hard to get rid of it. It goes and hides in the DNA, and so you can't get rid of it. We also have in our body these things called retrotransposons, and we're just starting to understand how they work. But there's this thing called Lyme-1 element, and during embryogenesis, it's actually very active, and in the brain, it actually causes a lot of recombination of our DNA. So the idea is that this is part of getting genetic diversity within the human genome. Obviously, this is also a potential cause of cancer because whenever we go and edit the genome, this creates problems. So in the context of gene editing, you probably potentially heard of CRISPR. And this is actually really, Jennifer Doudna discovered this at Berkeley, and this really opened the door to a lot of activity. And the idea is that you have a genetic mutation. We have essentially a technology called CRISPR that can recognize that, cut the DNA, and you can fix it. So this kind of idea is not actually new in the context that there are actually approved products. There are products at CSL, for example, that they use, but the most recent approvals were in sickle cell disease. Now, the problem with all of these technologies today is they generally have serious limitations. So if we focus on Casgevy, which is the sickle cell disease therapy that Vertex and CRISPR are developing, is a full bone marrow transplant, right? So you go through the full conditioning regimen. People have died from that. The cost is around $4.5 million Australian for the therapy, so it's not exactly accessible. Most sickle cell disease is in Africa, so obviously we're not going to be lining up many countries in Africa for that treatment. And obviously the other problem for these technologies, they're only available for simple genetic disorders, so very small mutations. If you wanted to, for example, cure cystic fibrosis, where CTR is a gene that is commonly mutated, the problem is that mutation isn't in a specific spot between patients. There's often different mutations within the gene. So the fastest way to cure that would be to just replace the whole gene. Otherwise, you'd have to personally figure out where each patient's mutations are, make a therapeutic product for each one. And so that's what we said we should do. We said we should create an RNA technology to address these limitations. And I don't know why we said this, because this has actually been the hardest thing that we've done. But we have, and we've recently started this company. And really the whole concept here is to address these limitations. Ex vivo manipulation. This is a problem. It's very hard to make cell therapies. Anything you have to do manually, it's not only expensive, but it really limits the ability to get your drug to where it may be most effective. You know, you don't want to have to live next to RPA to get a therapy out there. Delivery mechanisms are a problem. Genetic instability, we're seeing with lentiviral-based, so HIV-based technologies, that it's creating problems. There's off-target effects, and I already touched base a little bit on the size limitations. It's easy to correct 1 or 2 mutations in a gene. We can actually change up to about 20 pretty simply using prime editing. But most genes are anywhere from 5,000 to 14,000 nucleotides. Right? So being able to change 10 nucleotides, it's a good start, but it's not going to get us to where we want to go. And so what actually happened was I was starting Myeloid Therapeutics and we're using RNA, and you'll believe it or not, the investors all told me you can't do anything with RNA. RNA is useless. It's transient. You know, you need to be able to do this using a stable transduction system because this is a time before COVID, right, when RNA was invented. And so we actually, my team, we went onto the internet and we said, exactly what we did, RNA gene transduction. And this thing came up. It's called Lyme1. It's a natural retrotransposon in humans. And for those of you who aren't scientific, even I struggle with these slides. So you shouldn't worry about it. What is really important, I think, is it's part of that 80% junk in our genome that I told you earlier I was told was not worth learning. And what's really interesting about it is 2 proteins. It actually takes RNA from the cytoplasm, brings it back into the nucleus, and it has its own reverse transcriptase. What does that mean? It actually takes RNA from the cytoplasm and brings it back into the genome and writes it back into the DNA stably. So imagine if you could take this. It's an RNA program. You could take it, and you could actually engineer it to deliver the gene of interest. And so that's where we started in 2019. And it has been a hard slog to get to where we are. But we have finally, I think, cracked the code to this. And we've been very fortunate because we've attracted some investment around it. But essentially what we did is we took this endogenous retrotransposon, which is made of 2 proteins, ORF1 and ORF2. And we essentially took the very front of it and we chopped it off. And we put in an enzyme that can recognize the DNA exactly where we want it to drop in. And that's essentially now how we actually home into the DNA where we want to drop it. And on the back end of it, we put our gene of interest. But what we do is we put it in inverse, right? Because it's going to be reverse transcribed back into it. And the other thing that's really interesting is, unlike other technologies, the limitation on making RNA is not size. So we can actually make 14 kb size. So we've now overcome the other limitation of these other technologies, at least theoretically, right? And we started, at very, very low transduction levels. And Chris from CSL is here, and he'd look at this and be like, "You've got a long way to go." But you all start somewhere. And Prime Editing, who published in Science, their very first paper was 0.01%, and that was back in 2019. So we're on the right path. And in the last 3 months, and I put this in here specifically to point out the speed at which this field is moving, right? In 3 months, we've been able to get 10-fold increase in the production level using this completely RNA-based system. We can make this for $200. It's $4.5 million to get Casgevy today, right? So the whole pharma model that everyone complains about is, oh, it's so expensive to get therapies, right? But what's going to happen, instead of using social engineering to try and figure out how to get access to these therapies, we've got to focus on innovation, because innovation is how everything will become more cost effective. And I can summarize this a little bit on the next slide before I get on my soapbox about manufacturing in Australia. So essentially today we have 5 or 6 different approaches for gene editing. We have Cas9 nucleases, which is this CRISPR-based technology that Casgevy uses. We have base editing, just going to take 1 or 2 nucleotides and change it. Cas9 with integrases, I'm not going to touch base on that. And then prime editing, which can do up to 20 to 75 bases. What I'm focused on with this technology that we're building is putting a 14 kb, 14,000 length, nucleotide length gene into the nucleus of cells. And fortunately, I'm very happy to, be able to say that we've just got a little bit of seed capital recently to move this to the next level. Okay, so we've talked about immune programming, we've talked about editing of RNA, and we've talked about how we can do full genome editing. I'm going to end now on how do we stop designing this in America, design it in Australia, make it in Australia, and test it in Australia. And the reason I left Australia in the first place was because I had an idea as an autoimmune therapy, and I couldn't get any support for it here. And this morning, the company that I started announced another $1 billion deal with Genentech for another autoimmune disease. It's actually the 4th deal they've done in the last 9 years. And I was 26 at the time, and I went around and talked to all the venture capitalists. They kind of looked at me like I didn't know what I was doing, and I didn't. They weren't wrong, but it would have been helpful to have been told, you don't know what you're doing, but here's the path to take. So in Australia, we have historically a poor translational operational component to our system. We have great researchers. We have the best researchers in the world. I don't know if it's because we're so far away and it's the frugality and our culture, and it's about getting as far as we can and going back to that mentality I told you about, David Campese and Twickenham back in the day. I don't know if that's it, but there is definitely something about our researchers in Australia. And I'll take one of those any day over anyone else. But we haven't got a coordinated plan for how we solve for the issue of how do we take these great researchers, great science, and translate them. And, I'm just going to point out 2 examples. There's so many that we won't talk about. Wireless internet. But I will focus on things like CAR T cells. Right? It took 4 years. This was approved in the United States 4 years before it was approved here. mRNA vaccines. So the whole reason I even got engaged with Hugh was when COVID started, there was no component of how to make an mRNA vaccine in Australia available at all. Right? It's one thing to embrace globalization, when the whole world is on board with that. But when the rest of the world starts to become isolationist, you need to quickly shift your perspective. And I think in Australia, we're great comrades. We love to be on the same team as everyone else, but I think we have to be very careful about the path the world's on. And so I got to talking with different states here about, look, let's manufacture RNA here. And long story short is, I got to meet with Hugh. And so one of the things that Hugh was interested in, and I think he deserves and his team a significant amount of credit, is how do we build ecosystems, right? We're talking about RNA today, but if you think about chips and semiconductors, these are the other areas of national security interest, in my opinion, that Hugh spends a lot of time on. And I'm super interested in this ecosystem component. The key question is, what does it even look like? How do you build an ecosystem where you can have great academics doing great science? How can it be translated into therapies or products? And at the end of the day, it's all about money. I hate to say this, but it is, right? And what actually happens is success begets more success, and so on and so forth. And in Australia today, we're great at discovery, and we're great at phase 1 clinical trials, right? Everything I showed you from a clinical trial perspective, the very first person in the world was dosed in Australia. Australia is actually attracting more international phase 1 clinical trials than anywhere else in the world. It's because it has a deregulated phase 1 approach. You work with the hospital, the doctors, you don't have to go to the TGA. And I think this is actually very, very smart. But you can see here, if we look at the ecosystem, we have a lot of white areas that we're not necessarily very good at. And so the point is how do we change it? And I think if we think about what the key ingredients for an ecosystem, obviously vision, you need visionaries, you need money, lots of money. I told you how much we've spent, so this is going to be always a problem. Ideas, but one thing is advanced manufacturing facilities. It's all well and good if the next greatest idea comes out of the University of Sydney, but that academic has to go to the United States or to Europe to make it. It's already left the shores, and unfortunately, it's probably never going to come back, right? Which, to be perfectly honest with everyone here, I'm okay with. We have to also appreciate the fact that Australia is a small market. But the point is that if we build the ecosystem here first, and it doesn't have to leave, the time that these ideas have to leave Australia is the time when they command maximum value. And Australia reaps not only the value of having advanced therapies here, but it reaps the value of the economic impact of that, which today we're not doing. So advanced manufacturing is important. Train professionals and entrepreneurs. We have folks from New South Wales, with Andrew from UTS. These are great academics that are focused on training. How do we train these folks? This is really important. And in the context of advanced manufacturing, obviously we're talking about RNA. I showed you a slide earlier with all the increase in clinical trials. This is obviously an area of very, very important to me. So what happened? I met Hugh, and we actually had a building designed when I came to meet you. I said, Hugh, this is what you need to do, right? We need to do Moderna Light. So in Massachusetts, in Norwood, Massachusetts, just outside Boston, Moderna built the very first building in the world where all of the components, I'm going to touch base on this in a moment, are under one roof. And this had never been done before for a number of different reasons. It's very technically challenging because there's a number of different components that go to make it. And so I actually went on the internet to figure out who designed the building for them and found them and got them to design what I call the Moderna Light building, which has gone through a number of iterations now. And I met Hugh, and Hugh said, look, this is what we want to do, right? We want to do something here that isn't like building Moderna facility in Victoria. We want to do something that's accessible for academics, for startup enterprises here, and there's no strings attached, right? If you come to the building and you pay for it, you make it, you go. It's very simple. There's no equity component. You don't have to give any pound of flesh, which you have to do if you want to use other facilities around the country. And so we did that, and we've been doing this for a lot longer than I would have hoped you to get to here. We could work on that. But what's really cool, the first place in the world, right? So what's going to be at Macquarie Park? And I have a picture of it since you wasn't able to show it to you a minute ago. Analytical and process development support. This is really important. We're talking about next generation analytics, right? You take out a measuring stick to figure out if you want to cut a piece of wood, how long that is. We have to know how long a piece of RNA is. You can't even see it, right? We have to have very, very defined tools and technologies to do that. Plasmid production, this is important. You use E. coli to do it. The RNA itself is an in vitro process, has no bacteria. This is actually the key challenge. These are the things that people don't have to put in the one building because E. coli like to grow everywhere. And then LNP itself and fill finish. So all of this is going to be under one roof over in Macquarie Park. There's no roof on it yet. The wall's going up, I heard. But what's really important about this building and the approach that we're taking, I got to build a business. I have shareholders, and so we 100% are a capitalist enterprise. But you know what happens when you bring the really smart people from the academic institutes in Sydney to the table here, when you bring government to the table, we can actually align everyone's objectives. Because at the end of the day, the only way this is going to work is if we find really smart people to run it, really smart people to operate it, really smart ideas to put through it, and we build therapies that work. And I don't know if anyone's heard of a company called Vertex here. One drug made that multi-billion-dollar company. It was a cystic fibrosis drug. Gilead, one drug, HIV. These are household names in the pharma business, but it only takes one success, and that will drive all of this. And if it takes 10 or 20 to get there, then that's what we have to accept. We just have to accept that. It's a 20-year path. We have the choice to take it, or we can just worry about the next 6 months and we were having a conversation earlier with someone in the crowd about companies worrying about the next 6 months. It's always a concern. But we have the ability to build something really significant here, and Australia's size actually works in its advantage. Because we're small, because we're in a bubble, we shouldn't forget we're in a bubble, we can actually come together and do things that other people can't. And I think hopefully as we continue to progress, we can focus on that. So I'm going to wrap up now, almost perfectly on time. Hopefully, you can appreciate that, RNA is really fundamental, not only to life, but it has a number of measurable opportunities for us to use it therapeutically and beyond. And if you leave here, you should feel very optimistic about what we can do in Australia and in New South Wales. Take advantage of that. And the more we work together to push those goals, the further we will go. So thank you.
It was truly inspirational. I have to say, as an engineer and a kind of, someone who's interested a lot in physics, I found the last 3 or 4 months, as my various people in my office will know, I keep on reading these books on non-coding RNA, and I'm getting more and more enthusiastic about what you can do. We have some time for some questions, and I think there are some microphones going around. Come on, everyone, down here.
Susan Pond, excuse me, haven't spoken for a while. There was a Nobel Prize awarded for small interfering RNAs, probably in the early 2000s, I think. How do they play into this these days?
It's all part of it. So the siRNA, the little oligos, this is an area that's booming as well. So this is another system that you can use to modify gene expression, for example, by blocking things. So it's actually, that Nobel Prize was from folks at Northeastern University in Massachusetts. And so, that's just another use. So Alnylam is a company that does this. Wave Therapeutics, who's doing the other alpha-trypsin 1 ADA editing, also has a number of those. So the oligo space, it all plays a part in this. It's just not an area I brought up.
I think down here. So I think while getting there, I think the other thing that really interests me about this area is the use of digital technologies to do a lot of the design work, and also the fact that I guess, it's no longer doing one thing with one piece of RNA. Everything's kind of linked in a rather complex web and network of kind of ins and outs. Where are we going to go in that area? I think that's an area that Australia should have a go at as well.
So this particular data set here, so we have 2 products, right? One in TROP2 and one in liver cancer I mentioned. The same approach for TROP2 doesn't work for GPC3. So we take exactly the same non-coding regions for the GPC3 product, it doesn't express. So we actually used an AI algorithm to change the secondary and tertiary structure, plus the codons within it, and we incorporated a number of different modifications, and then suddenly it worked. So the answer to your question is all of these tools are going to be essential to optimizing where we're going, and I think things like RNA fold and all of these are essential to do it. I think we're actually working with ANU to try and build our own algorithm. At ANU they did some work where they took yeast, right, and they essentially expressed the exact same protein, but they took those non-coding regions and they made them different, and then they put the yeast in different stress conditions, and they figured out that different non-coding regions resulted in different expression levels of different proteins, right? No one really appreciated that non-coding regions were actually regulating what the cell would decide to translate and make, right? Now, think about that. Every single gene has a different non-coding region. The kind of computer power you need and biological repetition you need is a lot. So I think this is a massive area. I think this is uncharted.
But as I said, I'm an engineer at the end of the day, and you use computer-aided design tools to design stuff, and don't get me wrong, Saki here is bored of hearing me say, biology is becoming engineering, and it would be good to kind of, I guess, push that further.
If you're going to keep supporting what we're doing here, I'll agree with you.
All right. Might just go here, David. Sorry. David there. Sorry, go on. I apologise. I forgot.
That's okay, fine. You didn't mention blood cancers, and I'm wondering whether the RNA has any relationship to them or way of dealing with blood cancer. Sorry.
So in the context of diagnosing them or treating them, or both? So in the context of treating them, there are people that are in the blood cancer space using exact in vivo mRNA CAR technology. We just have not gone into that space. We're focused on solid tumours. There are a lot of people looking at extracellular RNA, so just in the circulation, in urine as well, for diagnostic purposes to see if you can find that, because obviously in cancers, aberrant proteins are overexpressed. So the idea is you'll be able to find these RNAs in the blood, in urine, a lot easier, and we have better technology, more sensitive to detect it. So that's an area of a lot of research right now in the diagnostic space as well.
David?
Yeah, can I take Hugh's question a bit further?
Last question, certainly. Yep.
A bit further. The whole engineering process is all about codification of process, right?
Yeah.
And I'm not hearing from what you've said that you've given us examples where each thing is very, very different. How much of the processes you do there are kind of stock standard and are going to be used, or do you still need a lot of brilliance to make any breakthrough at all? Like, where are we at with it?
Yeah, so that's a good question. I mean, I was trying to give a variety of different examples, but if we just take manufacturing, for example, most of these technologies will use the same underlying manufacturing approaches, right? In vitro transcription translation, for example. In the context of codifying, non-coding regions, we looked at over 1 million different combinations and permutations using algorithms. Then we tested 200. So that was kind of how we codified that. It's a very research-based question versus, when we're scaling up manufacturing, we're trying to build processes that are somewhat stock standard. So if you have an idea you want to do A, it's not a big stretch to do B, and so on and so forth. But there were definitely sacrifices we had to make in the facility. One of them is synthetic oligo manufacturing. We're working with Parley to build that up because the facility, we didn't have the resources to do both. So there are areas where certainly there's this codification going on. There's probably more areas we need to do it in. It's just still early. And most of it's run by biologists, not engineers. So this is part of the problem.
What could possibly go wrong? Right. Let's, if we could, again, thank Daniel for flying all the way out here to give this talk. I might just mention we've swapped our books. You may notice we give every speaker a book. The book this time round, I don't want to spoil it, but basically is a new history of the Royal Society of New South Wales. And I can highly recommend it to people. Really interesting read. And if you go back to with things, the Royal Society played an instrumental role in really getting the economy of New South Wales underway. So take a read. Thank you again, Daniel. Second. That's the last talk for the year. The first talk of next year will be on the 12th of March, and as by tradition, it'll be given by the winner of the Premier's Prizes for Science this year, Helen Christensen, and it will be on basically why are rates of youth mental ill health rising so rapidly, and what can we do to reverse this trend. So I'll see you all next year. Thank you very much. Thank you, Frank. That was a really interesting talk. Really.